Welcome to Unit 3.2 on Cell Structures and Function. I'm Wendy Nofke and I'm going to be doing your presentation today. The plasma membrane is an organelle that's common to all cells and creates the physical barrier to the outside world. Its unique structure creates selective permeability because it is composed of a phospholipid bilayer. What this means is you're going to have a phospholipid molecule that has a phosphate head represented in the picture by the spheres, attached to two fatty acid tails that hang down. As a bilayer, you're going to have the phosphate heads on the innermost surface inside the cell and then on the external surface of the cell. You'll have the hydrophobic tails that are going to be in the middle of this phospholipid bilayer. This is going to allow there to be watery substances inside the cell and watery substances outside the cell, but the two will not generally mix because they do not have to want to pass through those hydrophobic tails in the center. Cells will also have extensions and accessories on the surface. The flagella is a whip-like tail that's used for a swimming type of motion, seen in the picture on the bottom right. A common example of a cell with a flagella is the human sperm. The cilia are going to be hair-like structures on the surface of a cell. These are going to occur in eukaryotic cells, and they will have multiple of these on the surface. This allows for a crawling type of motion, or an example would be cells in the human trachea that are going to help to move mucus up to prevent that mucus and what is caught in the mucus from accumulating down in the lungs. Glycoproteins are molecules that are also found on the outside of the cell. This is going to help for identification and communication. Glycoproteins allow you to be able to recognize your own cells and to know which type of cells you have in your body where. Inside the cell we have the cytoplasm, and it's going to have a cytosol, which is a watery gel-like substance that's going to be inside the cell that will help to suspend the organelles. It also helps to maintain the cell shape and create a site for biochemical reactions to occur. The nucleus is the largest organelle in most cells. It's going to be the control center that contains most of the DNA. The mitochondria are another organelle that's involved with energy production. This is going to allow the cells to generate ATP, which is what it uses as an energy currency, from organic molecules like glucose or sugars. According to the endosymbiont theory, we have mitochondria in our cells because an ancient prokaryotic cell ended up engulfing a smaller prokaryotic cell, which eventually lost its ability to survive on its own, but started to function as the mitochondria in the cell. There's evidence of this because the mitochondria actually have their original plasma membrane and another membrane around it from being engulfed. They also have their own separate genetic materials, and they do have ribosomes that are more similar to prokaryotic cells. The endoplasmic reticulum and ribosomes are also important. The endoplasmic reticulum is used for transport. It has two varieties, the smooth and rough endoplasmic reticulum, or ER. The SER, smooth ER, has no ribosomes on the surface, but it actually uses bits of the membranes to form vesicles in the cell. The rough ER has ribosomes on the surface. Ribosomes are the organelle that's used for protein synthesis. The Golgi apparatus looks a bit like a stack of pancakes in the cell. This organelle is going to help to process proteins and prepare them for transport either inside or outside the cell. It's going to help the cell know where do I need to send this to. The vesicles and vacuoles are sac-like organelles, and we have a variety of these. A couple examples include the lysosomes and peroxisomes. Lysosomes are going to be vesicles that are going to contain lysozyme, a digestive enzyme that will allow the cell to digest foreign matter. Peroxisomes are going to use oxygen to help process toxins in the cell. The centrioles are a small organelle that's used to help to organize the chromosomes during cell division. The role of the centrioles is not completely understood, and we know that they are not essential for all cell division because these only occur in animal cells, and we do see cell division in plant cells without the presence of centrioles. Plant cells do have a couple of unique structures. They have a cell wall which is rigid and surrounds the plasma membrane. This is going to provide additional support and protection for the plant. While the larger animals have skeletal systems that help to allow them to stay upright and support their size, plants do not. So plants are going to rely on these cell walls to help provide that additional support for their taller structures. The central vacuole is going to store substances and help the plant to be able to hold its shape. Plastids are going to be structures that are going to have their own DNA, and these developed according to the endosymbiont theory, much like the mitochondria, in that they were a primitive prokaryote that was engulfed by another prokaryote. They also have their own DNA as well as the double membrane. One type of plastid is the chloroplasts. These contain the green pigment chlorophyll that's used for photosynthesis. Leucoplasts are another one that are going to store substances such as starches or be used to make small molecules.